Welcome, everyone. So first off, let's make sure that everyone can hear me. It's much more important that you hear me rather than you see me, because it's really the voiceover PowerPoint. So there, yes, I'm getting lots of yeses. So remember, sometimes I'm a little technologically challenged and don't push all the right buttons, unlike Mary, who's a pro at this. But anyway, I have Maggie in the background and so on and so forth. So what's our job to start out with? Well, most medical curricula basically start with anatomy, so it's kind of appropriate that we do the same thing. So I'm actually a neuroscientist by training, and I've taught basically anatomy review courses for Caplan for longer than I care to admit, but that's okay. So what we're going to do tonight, just to start out, is really deal with the most challenging of the anatomic sciences. Because as you all know in medical school, anatomy is really four different things. Anatomy is gross anatomy, it's neuroscience, it's embryology, and it's histology or cell biology. So we're going to deal with arguably the most challenging of the anatomy subjects and spend literally 4 nights just talking about central and peripheral nervous system neuroscience. All right, so when you took neuroscience, basically one of the things that probably drove you crazy, at least for a period of time, I've printed right here in the middle of the slide, and basically those were the tracty things, And depending on where you took neuroscience, the tract things, well, let's see, there were always something spino and spino something, and they were always kind of going up and down the brain, and some of them crossed and some of them didn't cross, and somebody was making you memorize 10, 15, 20 of them. Well, when you asked, well, what's a tract do? Lots of times the answer was, we're not sure, but we want you to memorize it anyway. So what we're doing basically when we review anatomy, any subject in anatomy, this is not really a review of all of anatomy or a review of all of neuroscience. It's a review of what tends to be tested on step 1. And over the years, we've kind of gotten, obviously, much information about what's tested on step 1. Certainly now it's all computer-based. And obviously, your exam is not going to be similar to anybody else's exam because of the nature of how the exam is structured. But the tendencies and the topics and the emphasis are basically always there. So my job is to review the elements of neuroscience in such a way to make you very comfortable with answering basically questions about lesion sites, causes, effects, pathologies associated, obviously, with central nervous system neuroscience. Because other than that, there are lots of neuroscience details that are out there that you probably had to learn in your neuroscience course in medical school that are never tested on step 1. So really my job, as well as the job of all of the other faculty that you will come in contact with, is to give you a level of emphasis and steer you through basically what we would call the overused term high yield topic matter that's tested on step 1. Okay, so in order to do this, we're going to use the anatomy notes, and you'll see at the top of basically up here at the top of the page, that I've indicated that this is page 247. So hopefully I've been able to copy the appropriate page every place that I have needed it. And with that page number in mind, we're going to go through the pages that are relevant. In some cases, I will point out topic matter that's important, or a way to remember it, or a chart, or a figure. I'm certainly not going to go over every page in the notes, but I'm going to go over many of them in some sort of order, basically, as we start, obviously, start our conversation about central nervous system neuroscience. Now, if you have questions, basically, and you think it's a question that might be relevant to the group, you can type right into the public chat, and I can sit here and read it, basically, and if I have time, which I usually do, I love to answer the question. So if you want to have a more detailed conversation or a private conversation, then you can certainly type your question in the Q&A and have a private conversation with our content TAs that are basically around the world that are obviously supporting everything that we do in terms of every subject going forward. Okay, so with that in mind, Maggie, can you pull that first slide away and let's actually look at the beginning of this PowerPoint. Basically, it does begin on page 247, section 4 of the new notes. And there it is. So First off, let's lay some groundwork about the nervous system. Some basic groundwork about the nervous system. Because again, we want you to have the basic concepts in mind and be comfortable with them, not just memorize them. So first off, on page 247, let's talk about what the nervous system really means and what its major components are. Certainly, the one thing that we can distinguish going right out of the blocks is, what is the central nervous system? The answer to that is the central nervous system consists of the brain inside the skull and the spinal cord. That's the CNS. And what we will see in a moment is that when we refer to the CNS, basically we always refer to it developmentally as being developed from the neural tube. So the neural tube will always be synonymous, basically, with the term CNS. Then, the peripheral nervous system, which is interconnected, simply contains cranial and spinal nerves. And these cranial and spinal nerves have to come in or go out of the central nervous system. So they're interconnected. So these are two interconnected parts of the nervous system. Note basically how we organize the neurons and the processes in nerves. Number one, basically, nerves contain axons that grow out of the neural tube. And they will be basically skeletal motor axons and preganglionic autonomic axons. They will have their cell bodies inside the neural tube, and only their axons will be found in a peripheral nerve somewhere. The other major source of neurons that we find in cranial and spinal nerves are derived from something very, very important to all aspects of embryology, and that's neural crest. That's neural crest. Okay? So what's developing from neural crest will be all or virtually all of our primary sensory neurons and all of our postganglionic autonomic neurons. Okay? So, Hans, you're making a good point. So cranial and spinal nerves are basically part of the central nervous system and part of the peripheral nervous system. So we only technically find nerves in the peripheral nervous system, but they have to have components in both places, because nerves are sending axons into the central nervous system, and they basically contain axons that grow out of the central nervous system. So they are interconnected. Okay. So that's where our basic plan is going to be. So one of the first major topics we're going to do is related to embryology. Now, of the things that are tested on step 1 in anatomy, I can reduce it to the ridiculous. The things that are tested in step 1 in anatomy are issues, cases in the central nervous system, and nerve lesions. All of the peripheral neuropathies involving things like the brachial plexus and the lumbosacral plexus and cranial nerves, so on and so forth. One of the other topic areas that's heavily tested in anatomy is embryology. So I already know the results of the poll before we do central nervous system embryology. So this is obviously a test of the chat. Obviously type in the public chat, but I know the results of the poll. How many of you out there somewhere in the world liked embryology when you took it in medical school? How many of you? Yes or no? Did you like it? Did you not like it? What was your feeling about it? Yep, the results of the poll are about the same. There we go. Very few people basically like embryology. Maybe 10% of the world does. So step 1 must know that, because step 1 typically shows you or exposes you to lots of embryology questions. Now, obviously, they will deal with all aspects of human development, every organ system, okay, starting with the nervous system. So in order to do that, let's look basically and spend some time on the next page. This is page 248. So what we're going to do first, as our first major topic in neuroscience, is make a brain. And talk about not just how it's made and the factors that are involved, but certainly we're looking for the congenital malformations that basically are seen during fetal life that affect brain development. That's what the exam is going to ask you about. All right, first off, let's make a couple of comments, and you'll hear much more about what's called early embryology in a couple of weeks when you hear Gross and Embryo from Dr. Seiden. So, first point that we want to make is what happens in early embryology at a process that's known as gastrulation. What is gastrulation? Well, I typed it up at the top of the page. Gastrulation is a process that you'll hear about in some detail, but gastrulation is a process that establishes the three primary germ layers of the future embryo from which virtually all organs and tissues will be derived. In fact, we can argue that most organ systems basically contain at least two out of the three germ layers, in some cases all three. Okay? So gastrulation is a process that establishes the three primary germ layers during development. So basically, let's look at them over here. This is a section right here through the developing embryo, very early, about day 18. And what you're seeing here in the section at letter A is basically the three germ layers stacked right on top of one another. Note that the outermost germ layer in blue will be ectoderm. The middle germ layer that's kind of brown or tan is mesoderm. And the innermost of the germ layers is endoderm. Again, we'll be very specific about what these germ layers form when you get to other organ systems. But tonight we want to concentrate on this fact that the entire nervous system, the entire central and peripheral nervous system are derived from ectoderm. So the entire brain and the peripheral nervous system will be formed from basically one germ layer, which is unusual. But what is the other thing, the other major organ that's going to be formed from ectoderm? The other major organ that's going to be formed from ectoderm is skin, the largest organ in the body. So in essence, what we're saying is the entire brain is forming from the same germ layer that will form basically skin. Okay? So obviously, we have to have a process that separates the developing nervous system from this germ layer, obviously, so it can internalize itself and be found invested by bones of the skull and bones of the vertebral column. Note, basically, during the process of gastrulation, a very important rod-like structure forms in the midline in the underlying mesoderm. And this rod-like structure is illustrated on here is known as the notochord. Notice the notochord. Basically, the notochord gives rise to signaling molecules and transcription factors that induce the formation of the nervous system by precipitating changes that occur in the overlying ectoderm. So one of the major inducing molecules that's secreted by the notochord is something called sonic hedgehog. That plays a major role in inducing changes in the overlying ectoderm. So, saying this again, basically, the entire nervous system will develop from a subset of ectoderm, the layer in blue that we see here in this section, and that same ectoderm layer will actually form skin. So, and what induces changes in the ectoderm layer to form the nervous system are factors produced and secreted by a rod-like structure that develops basically in the underlying mesoderm called the notochord. And we pointed out the notochord basically on this slide as you see here. So first off, where does neurulation take place? Where is the first place in the human body where gastrulation forms these three germ layers? The answer is, if I were to turn around, basically what we're looking at in this view over here on the upper left corner of the slide is the future dorsal part of the future embryo, the future dorsal part. Because we have very little craniocaudal polarity, and we certainly don't have any limb buds yet. But basically, gastrulation first establishes these three primary germ layers in the future back, the future dorsal part of the embryo. And this is also where the entire brain will develop. It will develop basically in the midline endoderm that in essence is parallel to the vertebral column. Okay? So the process of gastrulation takes place during week 3 of early embryonic life. And it marks the beginning of what we call the embryonic period. Good question. All right. So what is the first thing that the notochord inducement actually does? It causes the midline ectoderm, lying in the dorsal midline, to thicken first and form a neural plate. Basically, that's just a thickening of ectoderm. And once it thickens, we now refer to it as neuroectoderm. It's a subset. So then, under further induction, the next thing that happens is we start off with a thickening of ectoderm called the neural plate. Then, under further induction, the midline portion of the neural plate invaginates and forms a shallow neural groove flanked by a pair, as we see on this slide at the top here, section A, of neural folds. So the neural groove gets a little deeper, the neural folds get a little more elongate. But now, what is the major event in neurulation? We started out with a neural plate. We developed a neural groove flanked by neural folds. What's the major event in neurulation? It occurs when the edges, the open edges of each of the neural folds migrate toward the midline and fuse, forming a hollow neural tube. As we indicated on our introductory slide on the previous page, the neural tube will form the central nervous system. Okay? But now, look what is also developing adjacent to the edges of each neural fold, also from neural ectoderm, a special group of cells called neural crest. Note that the neural crest cells basically are shown right here in a little darker shade of blue that are developing adjacent to the edges of the neural folds. And note what happens when the neural tube closes. When the neural tube closes, it completely separates itself from the overlying ectoderm, and the remaining ectoderm grows over the site where neurulation took place. So there's no evidence postnatally that neurulation basically occurred or developed from the same germ layer that forms skin. And note, after the neural tube closes, it's not only completely invested by mesoderm, but look at the key location of neural crest. Neural crest cells, you can see, are developing, and this is a key point, outside of the neural tube. And originally, they develop in what we call this kind of this dorsolateral position that we see them here. And from their original developmental position, the neural crest cells will migrate, and they will become, as we saw on the previous page, primary sensory neurons by forming, Hans is correct, dorsal root ganglion cells and spinal nerves, and they will also form postganglionic autonomic neurons, meaning that any kind of ganglionic neuron, which is defined as a collection of cell bodies outside the CNS, will be derived from neural crest. Obviously, they're outside the CNS, they're outside the neural tube, so they have the opportunity to obviously make or form many of the neurons that we find in peripheral nerves. Now then, let's look over at the picture at the lower left corner of the slide. Because now we want to understand or distinguish what is the time course of neural tube closure. And if it doesn't close, how do we describe basically the neurologic deficits associated with that? Note, first off, it's kind of interesting and unusual in a way, that the middle of the neural tube closes first. The middle of the neural tube, maybe somewhere basically in the upper end of the spinal cord, lower end of the brainstem. It's closing first. And that's closing about day 23 of embryonic life, early in the 4th week of development, immediately after gastrulation, which was established during the 3rd week of embryonic life. All right, so then note that the rostral or the cranial end of the brain, which certainly forms the bulk of the central nervous system, closes second about day 25. And then look what closes third and last. The caudal-most portion of the brain, the caudal neuropore, closes third and last about day 27. So it's over a sequence of days during week 4 that the entire neural tube closes. The middle first, the rostral second, and the caudal third. So now, basically, if either the rostral neuropore or the caudal neuropore fails to close, the fetus has what we call an open neural tube defect. Open neural tube defect. And I'm emphasizing the word open because, as we will see on the next page, there are also a number of closed neural tube defects that we need to distinguish. So certainly open neural tube defects are not going to be compatible with life because you can't survive without basically a part of the brain that fails to develop by initially closing. So now let's put some markers on it, some screening markers that might be evident during the pregnancy. If a fetus has either a rostral neuropore defect or a caudal neuropore defect, what's going to be elevated in maternal serum and also in amniotic fluid? Alpha-fetoprotein, the most prevalent protein basically produced by the fetal liver. It's a form of fetal albumin, if you will. So, and it's thought that if there is a neural tube defect, in essence, it's a body wall defect. Because remember, the entire neural tube is developing from the surface layer of the body wall. Okay? So basically, we're going to see a number of examples in embryology where amniocentesis or maternal serum sampling will show elevated levels of alpha-fetoprotein. So they're pathognomonic for open neural tube defects, but look at the note on the lower right corner. Elevated levels of alpha-fetoprotein basically can be elevated with what we call ventral body wall defects, such as gastroschisis or an omphaloceal. So just because an amniocentesis shows elevated levels of alpha-fetoprotein doesn't mean that the fetus has a neural tube defect. So these are ventral body wall defects over here that basically show up, obviously, with elevated levels of alpha-fetoprotein. So alpha-fetoprotein will be elevated in neural tube defects, which are body wall defects, dorsal body wall defects. Ventral body wall defects, two examples of them are gastroschisis and an omphaloceal. And Dr. Seidnow distinguished between the two pathologically, but they both are ventral body wall defects. However, there is a second marker that is unique to just open neural tube defects. And that additional marker is elevated levels of acetylcholinesterase. So basically, if not only alpha-fetoprotein is elevated, but also acetylcholinesterase is elevated, that's an absolute double marker for just open neural tube defects, such basically as a rostral neural tube defect or a caudal neural tube defect. So we need some terms for these. Basically, and we'll get into the closed neural tube defects, Jeffrey, but basically with regard to that. So first off, let's look at how we describe basically open neural tube defects. If the rostral neuropore fails to close, we refer to that as causing anencephaly. Anencephaly, yes. So anencephaly is a rostral open neural tube defect. And basically, we're seeing it here. Then, basically, what is the classic open caudal neural tube defect? It's known as spina bifida, but it's the most severe form of spina bifida, which we'll see on the next page is known as myeloschisis. So only these two defects, which are open neural tube defects, will present with both elevated levels of alpha-fetoprotein and elevated levels of fetuses basically that have open versus closed ACHE. Whereas let's look at our newly formed brain. Okay? So a dorsal body wall defect is a neural tube defect because that's the only significant thing that's developing in the dorsal body wall, neural tube defect. All right, so here's our newly formed brain. And we see it basically, there's the neural tube right there. There's the cranial end of the neural tube. There's the caudal end of the neural tube. So the point that we want to make is that the cranial end, the rostral neuropore, undergoes significant elaboration by initially forming 3 primary brain vesicles that then further differentiate into 5 secondary brain vesicles. And it is from the 5 secondary brain vesicles that all parts of the central nervous system, except the spinal cord, will develop from. So in a way, we're going to introduce our entire content this week by identifying the parts of the postnatal brain, by simply illustrating what the secondary brain vesicles form during development. So first off, note that the largest of the secondary brain vesicles basically is the telencephalon, and certainly the major thing that it forms are a pair of cerebral hemispheres, one on each side. Note also that most of some motor control structures that we'll talk about in a couple of nights, known as the basal ganglia, are forming from the same secondary brain vesicle, the telencephalon, as the origins of the cerebral hemispheres. Then, basically, note that the next part of the postnatal brain forms the diencephalon. And here's basically where we're going to get a little help. The telencephalon basically is the top dog. Largest part of the brain develops from the telencephalon, the cerebral hemispheres. The diencephalon, this is a key memory point for you. The diencephalon gives rise to 4 brain components that all end in the word thalamus. For example, if you look down at the chart down at the bottom here, you can see that anything in the diencephalon ends in the word thalamus. So let's animate this briefly, just to give us an introduction about what these thalamic components are, which we'll talk about basically on a subsequent conversation. So first off, I'm just going to animate this. What is the largest component of the diencephalon? It's the thalamus. What does the thalamus do? It plays a major role in relaying all subcortical information to specific areas of a cerebral hemisphere. Specific areas of a cerebral hemisphere. So we're going to be very specific as we build neural systems. Yes, both sensory and motor information, absolutely. We're going to be very specific about the names of thalamic nuclei and which sensory and motor systems are being relayed through that nucleus to certain parts of cerebral cortex. Okay, so that's the largest. Then the second largest component of the diencephalon is called the hypothalamus. Hypothalamus is playing a major role in regulation of all of the body's endocrine secretions, largely by controlling the activity of the pituitary gland, which is suspended below the hypothalamus. And we'll also see in a bit in our next hour that the hypothalamus plays a major role in controlling autonomic function. You've got to figure out how it does that and basically what's significant about that. Then, the third component of the diencephalon basically is a fancy word called the epithalamus, but that's really the pineal gland. And lastly, the subthalamus is really a part of the basal ganglia. So the bottom line is basically it has a number of diverse functions, obviously related to much of neural processing that's going on in very different areas of the brain. Last but not least, look what is written down at the bottom of the list under diencephalon. The words retina and optic nerve. Note that everyone knows that the optic nerve is always referred to as cranial nerve number 2. But basically, starting right now, I'm reminding you that the optic nerve is not a nerve. It is a tract of the brain, meaning that it will be surrounded by the same meningeal coverings as the rest of the central nervous system, and the optic nerve basically is never outside of the brain. And basically what we say is the optic nerve is an outgrowth of the diencephalon, because when the optic nerve projects back into the brain, back into the rest of the central nervous system, it will have major synaptic connections with parts of the diencephalon. Okay? So going forward, remember that the optic nerve is not a nerve, basically. It is a tract of the brain, meaning that it's going to have different clinical implications than any other cranial or spinal nerve going forward. All right, then, the remaining 3 secondary brain vesicles, basically, are forming the components of the brainstem. The mesencephalon forms the midbrain. The metencephalon, pons and cerebellum. The myelencephalon forms the medulla. Okay? So, yes, the olfactory are a series of olfactory neurons. Basically, they are in the peripheral nervous system. But they are actually, there isn't really something concrete as an olfactory nerve, because it's a bunch of little olfactory neurons that go through a sieve-like plate of bone from the nasal cavity into the central nervous system. Okay, then note again, and we'll play on this as we go forward, that the neural tube developed in the form of a hollow tube, and the neural tube maintains its hollow caliber throughout life. So what basically is in the hollowed out portion of the neural tube? The answer is ventricles and channels that connect one ventricle to another. You can see that every part of the brain contains either a ventricle or a channel that's connecting one ventricle with another ventricle. So now let's look basically at the details regarding defects that occur in neurulation. First thing I want you to do is point out or underline or do something, mark it. I can actually do it here. Is basically underline the word anencephaly. You can do it. Underline the word anencephaly. Right there. There it is. And go down here to letter D and indicate letter D, which is known as spina bifida with myeloschisis. Basically, anencephaly and myeloschisis are the 2 examples on this page of open neural tube defects. Anencephaly results when the rostral or the anterior neuropore fails to close. Myeloschisis basically results when the caudal end of the neural tube fails to close. So only these 2 conditions, a fetus that has anencephaly or myeloschisis, will have basically the 2 markers present. These conditions will show elevated levels of both alpha-fetoprotein in amniotic fluid and acetylcholinesterase, because both of them leak out basically at the site where the neural tube defects are the same. So these are the 2 open neural tube defects, and they are the only 2 that present basically with both markers being elevated. So certainly you're going to use ultrasound imaging, the gold standard, to go look for the defect. But suppose ultrasound imaging is controversial or not conclusive. Is there another way to distinguish a fetus that is anencephalic versus the fetus that has myeloschisis? The answer basically is the presence of polyhydramnios. So if a fetus is anencephalic, there will be elevated levels of amniotic fluid, potentially complicating the pregnancy. Whereas if a fetus has myeloscus, amniotic fluid levels will be normal. Why? Because if the rostral end of the brain doesn't form and we have a fetus that has anencephaly, there is no neural mechanism for swallowing. Okay? So only in an anencephalic fetus will amniotic fluid levels be elevated, polyhydramnios. In a fetus that has myeloscus, we again will still have elevated AFP, elevated ACHE, but normal levels of amniotic fluid. Normal levels of amniotic fluid. Let's say this again. So only anencephaly and myeloscus, they are the two open neural tube defects that present with elevated levels of both AFP and acetylcholinesterase. But if a fetus is anencephalic and the rostral end of the brain is missing, there's no neural mechanism for swallowing, and amniotic fluid levels will be elevated. Whereas if a fetus has myeloscus, amniotic fluid levels will be normal. Okay? Yes, and Mario made a good point. The term myeloscus literally means split brain. Myelo brain, schism, a split. Good point. All right, now, what's shown down here at A and B and C are examples of closed neural tube defects. Closed neural tube defects. Why do I say that? Because look basically what's there and there and there. A normal appearing spinal cord. So in all three of these conditions, A, B, and C, the caudal neuropore closed and formed a normal appearing spinal cord. A normal appearing spinal cord. Okay? So, what's the problem? Well, the answer is there may not be one. So these three, A, B, and C, are the milder but more common forms of spina bifida. In fact, they're the true forms. Because what's common to all three, A, B, and C? In all three of these conditions, the newly formed spinal cord fails to induce bone to grow around it. So in all three of these conditions, there are missing spinous processes, where the two laminae that develop around the newly formed spinal cord fail to fuse and form a spinous process. So that's the true form of spina bifida. It literally means a vertebra with two spines, two laminae that fail to join together in the midline, basically forming a spinous process. So what's different about them? What's different between A and B and C? Yes. So basically, A, B, and C are normal spinal cords because they're basically closed caudal neuropores. So my fax machine is ringing in the background. Anyway, so what's different about them? The most common form of spina bifida is known as spina bifida occulta. What's the occult mean? Hidden, mysterious. So this is the asymptomatic form, where there are only a couple of missing lumbosacral spinous processes. Basically, this is not a body wall defect either, because ectoderm grew over the site where neurulation took place. So there's no elevation in amniotic fluid of alpha-fetoprotein, certainly no elevation in acetylcholinesterase. So all the fluid levels are normal. There's no cyst-like protrusion out where the spinous processes are missing. The only classic marker that's seen in this newborn is a tuft of hair over the site where the spinous processes are missing. So basically, this is the mildest but most common form of a closed neural tube defect, where there are only missing spinous processes, skin grows over the site where neurulation took place, and we simply have a tuft of hair marking the point where the spinous processes are missing. normal levels of basically all of our markers or screens during the pregnancy.
Good question. Nobody knows why there's a tuft
So
of
that's
hair
spina
marking the
bifida
site where spinous
occulta.
processes are missing, but it's classically
Then,
pathognomonic.
basically, nobody knows why. Good question. Nobody knows why there's a tuft of hair marking the site where spinous processes are missing, but it's classically pathognomonic for a newborn that has spina bifida occulta that had no recognizable elevation or changes in markers, basically, during fetal life. Then, what's going on with B and C? In both of these conditions, there is a cyst-like protrusion out in the dorsal midline where spinous processes are basically the bony dorsal projections of each vertebra. Spinous processes basically are formed when a pair of laminae of an individual vertebra join together. So in both B and C, there's a cyst-like protrusion lined by dura and arachnoid that contains cerebrospinal fluid that basically is herniating or evident out where the spinous processes are missing. But now, the presence of the cyst means that ectoderm could not grow over the site where neurulation took place, and in both B and C, there are going to be elevated levels of alpha-fetoprotein detectable in amniocentesis, even though we have a normally formed spinal cord. Because again, AFP basically is leaking out an amniotic fluid through a body wall defect. Okay? So for meningocele, yes, basically they're going to be a loss of skin or an absence of skin that's covering basically the meningoceal or the meningomyelocele. So then what's the difference? Basically, if the spinal cord is in its normal position, the condition that results will be a meningocele. If the spinal cord is displaced into the cyst, that's known as a meningomyelocele. The word myelo means brain. So in both B and C, the spinal cord forms normally, there are missing spinous processes, and alpha-fetoprotein leaks out and is elevated in maternal serum and in amniotic fluid. Okay? Because the cyst creates a dorsal body wall defect. Okay? So that's really the way to distinguish closed neural tube defects from open neural tube defects. Certainly the exception to everything, the least symptomatic form of spina bifida is spina bifida occulta, where skin grew over the site where neurulation took place, normal levels of basically the markers, and a tuft of hair marking the defect. But in B and C, again, closed neural tube defects, only AFP will be elevated during amniocentesis, not acetylcholinesterase. Because ACHE was only elevated in a fetus that was anencephalic or had the most severe form of spina bifida, which is known as myeloscus. So again, basically AFP is elevated when we have closed neural tube defects if we have a meningocele or a meningomyelocele that is in essence a body wall defect, allowing alpha-fetoprotein to leak out through the defect and into amniotic fluid where it will be elevated. Okay? Because remember, when we talk about neural tube defects, that's really a body wall defect. Okay? So that's basically the point. And then myeloscus, look at myeloscus, where we have the laminae of the neural plate basically situated on the dorsal aspect of the embryo. So only in D, myeloscus, and in anencephaly will we have elevation of both alpha-fetoprotein and ACHE. Correct, basically. There is no body wall defect in spina bifida occulta because the skin grew over the site where neurulation took place, basically. So yes, there's a body wall defect because of the presence of the cyst in both a meningocele and in a meningomyelocele. Absolutely right. So all we're doing is distinguishing basically the markers that we would see in different forms of either an open or a closed neural tube defect, and that's been the whole point of the conversation. Okay, so yeah, so alpha-fetoprotein is absolutely normal levels in spina bifida occulta. Absolutely right. Absolutely right. So let's see what Jeffrey's saying. So in all subtypes of spina bifida, there's no polyhydramnios. Yes, polyhydramnios will only be elevated in a fetus that has anencephaly, not myeloschisis. Because the caudal end of the spinal cord has nothing to do with swallowing. Nothing to do with swallowing. Okay. Aside from polyhydramnios, no. Well, we already have the markers. We already have the markers. If a fetus is anencephalic, you're probably going to see it in ultrasound, but you're going to have elevated AFP, elevated ACHE, and polyhydramnios. Okay? And basically, is there a difference between A and B? Yes, because B will have a cyst. Basically, will have a cyst. So spina bifida literally means missing spinous processes. Missing spinous processes that will be evident in A and B and C. But these are all basically, B and C are basically different because in C, the spinal cord between the two. Okay, so basically, let's finish this developmental story simply by looking on the next page and talk first about what is the Arnold-Chiari formation. What is the Arnold-Chiari formation? Okay, the one that has polyhydramnios is anencephaly. Anencephaly. Okay, anencephaly is the only one that will complicate the pregnancy with polyhydramnios because the rostral end of the brain is needed for swallowing. Good. So there are two forms of Arnold-Chiari. One much more common than the other. Type 1 is the most common. It's largely asymptomatic early on. It's caused by a downward displacement of part of the cerebellar hemispheres, and that can block basically the area of the fourth ventricle, specifically as the cerebellar tonsils, and it is frequently associated with enlargement of the central canal down in the spinal cord that creates something called syringomyelia, basically, that develops typically later. So it can block the fourth ventricle and can cause forms of hydrocephalus. Type 2, more serious, more likely symptomatic, and it's caused by a downward herniation of the midline of the cerebellum. The midline of the cerebellum, basically, down into the foramen magnum, and that typically is more likely to cause a form of hydrocephalus, and that type 2 is frequently associated with another caudal defect known as a meningomyelocele. So it turns out that the syrinx is more likely to be associated with Arnold-Chiari type 1, and the meningomyelocele more frequently associated with Arnold-Chiari type 2. Then, the Dandy-Walker malformation is literally a failure of one or more of the openings, basically, to develop in the fourth ventricle, again causing a form of hydrocephalus. We'll talk about the causes of hydrocephalus in a little more detail, basically, in the next hour. Then, basically, what is holoprosencephaly? This is a very genetically based neural tube defect. It's seen in trisomy 13, or the Patau syndrome, and it leads to incomplete separation of the hemispheres, basically, where they only have one ventricle and maybe even one eye. Okay? So both Arnolds have the potential to block the fourth ventricle. The type 2 is more likely to block, basically, openings of the fourth ventricle and cause hydrocephalus, although either condition might obviously result in that. So good place to take our first break now that we've made a brain. So good place to take our first break now that we've made a brain and talk about all the neurulation defects. So you can see the timer that's posted up by Maggie, the Tech TA. If you have subsequent questions, ask them. I'm not going anywhere. When we get back, we'll move on to other aspects of the nervous system now that we have made a brain.